get a sense of people's interest and also. So yep, we'll get started here. So I'm Bob Fulton from GTAC at MGI. We're going to tell you a little bit about Spatial. So Spatial, the next frontier. You can have the whole little Star Trek thing going through your head now, just like me. All right, so the topics we're going to cover today, technology selection, basically the process we went through to pick this and realize that this was on the fly. Some of these platforms were or were not available, so I'll talk a bit about that. Practical implementation, thinking about your experimental design, MGI capabilities, and then the future, especially from an informatics perspective. So I'm going to run through the platforms that we evaluated, and we looked at a variety of other things as well. And I feel like a paper was coming out about every other day for a while in this space. So this is definitely challenging to keep up with. But so VizGen, this is one of the early groups that we engaged with. And they have available platforms. So this will be, I'll go through the same common theme. They can do about 500 custom genes at a time, minimum of 20 slides for that. They have pre-designed panels that are about 300 genes. All these, and some of these numbers are a little dated, and they continue to grow. And those are in units of 4 and 12 because they're pre-made. One slide in about 30 hours with another 10 hours of analysis time, and it'll do a centimeter squared on a slide. They do have proteins available along with nucleic acids, six co-staining at a time with the nucleic acids, and there is a fresh and FFPE workflow. NanoString, the Cosmics, and the Geomax or Geomics was the prior iteration. So one issue with this, the new orders being delivered are from 2024. So if you ordered a fleet, you had the possibility of getting one early, but the availability of this platform was challenging when we were evaluating. Customs, this is not made for custom for the most part. Customs were a few $100s per, and you got 192 reactions. So you had to have a pretty well-defined assay for that to make sense. They have a pre-designed panel of 1,000 genes with 50 customizable in that space. They on their roadmap is 6,000, and they have data demonstrating 6,000. That's about a 9-day turnaround, and I'll talk a bit more in sort of a table form on the decisions and the timelines. 64-plex protein panel is available, but it's on an adjacent section. It takes a couple days to run that as well. But again, on the roadmap is to put that with the nucleic acids. Fresh and a frozen, or a fresh and a FFPE workflow again. And this can be coupled with the Geomax or Geomics platform. That platform does do whole transcriptome with a sequencing component on the back end. It's not single cell, and you'll hear this theme with the Visium as we talk about 10x as well. But the Geomics is not single cell, but you can outline regions based on morphology or just regions of interest in general. The Akoya PhenoCycler fusion system. So we did investigate this. This is more proteomics than nucleic acids. They did have available platforms. They could do 12 nucleic acids at the time that we investigated this, and they were on a trajectory to have 100 nucleic acids. One slide and 100 proteins along with that. Well, yeah, I'll get to that. One slide can run in hours or days, depending on the target space that you give it. And then 18 by 35 millimeter space, and again, proteins as well. And then 10x Genomics Xenium, and I'll save you the surprise. This is the platform that we ultimately went with. We installed the first right at the beginning of May, I believe it was 2023, and we had a second by June of 23. It does have a fresh and a FFPE workflow. So these are hot off the presses, actually. So the 480 custom gene panel, that is new. I think when we started this endeavor, it was about 350. And this is hot off the presses as of yesterday. There are also spike-in components of 50 and 100 genes that you can also order, and those can be standalone or spike-in on pre-existing designs. So the pre-existing design panel also took a leap to 480. Again, this is, you'll see in a slide or two later, where there's about 350 genes in many of the panels, 300 to 350. 2 slides in about 48 hours on instrument, and each of those slides does 12 millimeters by 24 millimeters. Try to think of this in centimeter cubes to just sort of normalize across all the platforms. So this would be 4 per 48 hours. Proteins in parallel are not currently available. Again, you could do an adjacent slide on a different platform, but not yet available, but on the roadmap. And the tissue, the post-xenium is still intact. So you can do subsequent assays on that tissue downstream, and Rich will talk a bit more about this. It can be coupled with the Visium for FFPE, which is a full transcriptome. So similar to the CosMx GeoMax sort of relationship, it's a similar situation here with the Visium and the Xenium. And then it can be coupled with other 10x genomics assays, and they have software to meld together data sets and make better use of all the data in full context. Okay, so here's the table that we made, basically trying to make choices. So some of the key attributes that we were looking at. Works better here. The availability, again, this was hot off the presses, both the Xenium. So the Xenium was tight, where trying to get that in-house. VizGen and Akoya did have an advantage where they did have available platforms that they could place immediately when we were exploring this. And again, NanoString, depending on your order size, most were being placed after the end of this year. Throughput capacity, again, was a key attribute that we liked about 10x. 2 of those slides in 48 hours, so roughly 4 centimeter squares in 48 hours. VizGen was 1 in 30 hours. Akoya was 1 in a varying period of time. One of the knocks on NanoString is NanoString took a different tack. Instead of a concise gene list, they went with the larger 1,000, moving towards 6,000, but that takes 7 days for 2 slides. And it'll do bigger than a centimeter square, but it's proportional. The space and time are proportional to each other. So if you want to do a bigger space, it can do it, but it takes longer. Protein space, again, this is where VizGen and Akoya shined. 10x, we have confidence that they're going to get there soon. And NanoString is adjacent now. They have an assay, but they're working towards the same slide as well. Company infrastructure, this mattered a lot as well. So we've been down this road before where you take this sort of hot new thing, and sometimes that comes with its challenges. So 10x has a long track record. We've been working with them for 10 years on, from a single cell all the way through to the various tools that they have. Good service staff, their website, their helplines, all of that is second to none in my opinion. They've been great from that perspective. VizGen, they're a small company. They were at the time about 100 employees, so that was a little worrisome. But again, they've been very supportive and interactive as well. So no knocks on any of these companies. They've all done a great job and have a great product. Akoya, again, pretty strong infrastructure. Again, they were just more protein oriented. And NanoString, the big knock is the timeline to get the platform. That was the challenge. Data management, I'll show you in the next slide. So I am not a bioinformatician by any way, shape, or form. And the data management for the Xenium in particular, I downloaded the Explorer software, and I'll show you some images that I generated in about 10 minutes. So that's pretty remarkable from my perspective. VizGen, they had clunky, like sort of big data files. It was difficult. They didn't have a Mac interface, for instance. There were just clunky. There was clunkiness to that, and again, large data files. Akoya, again, large data files, had reasonable software. NanoString is cloud-based, so you can go to the data. They did have competitive storage and processing models, but again, the data was sitting on the cloud. Other value added, the 10X, the integration of their various tools is really powerful, and Rich will talk a lot about that, I think. The VizGen Merfish, they were early and available, and again, worked with us very closely, and they do have proteins available with the nucleic acids. Akoya, again, strong proteins and an available platform. And then the integration of the GeoMax with the NanoString is a plus. So here's my views that I downloaded again, it literally took me 10 minutes from downloading the software. So this is just a cell slurry that was frozen, put in OCT, embedded in OCT and frozen and sliced and fixed. And what you see, that's 27 genes. So this was just to validate the platform. But there's 27 genes on here. I have them all turned on. So the colored circles there, primarily in the nuclei, those are the transcripts, and you can literally count them. The bottom left corner is the same, just zoomed in, and I just lassoed an area, and you can't see it here, but darn, that is delayed. There are like gene numbers. You can output the transcripts to make t-SNE plots, to all sorts of nice, quick, easy little tools. This is just a zoomed in view here. And again, you can see the transcripts in the cell, which of all the things, I've been doing this a long time, and of all the things, this just still amazes me. And then this is a first run of real samples from the Millman lab. And this is a mouse brain, and again, zoomed out, of course, but you can see the pretty data. Current and developing panels. So you see what's available now here on the left, and you can see the numbers of genes. So it looks like it ranges from about 250 genes to almost 400 genes. We got a couple more panels that are on the way here very soon, human skin and human colon. There are other ones that are in the works, and Rich will talk a bit more about that as well. So looking forward, near-term advances, development. So expanded target space. So this was a promise coming from 10X as well. And there's different schools of thought here. Again, NanoString took this tack early on where they wanted to be comprehensive about what they made available, but then it takes longer, and there's more molecular crowding, and there are issues with that. But you do get a more comprehensive picture. So 10X has adjusted to that a bit, where they were originally smaller panels. And they're going both directions, actually. So they're working on a 2 and a 5K panel. I've seen data from both, and they look good. So they're looking at the first half of next year for that. They've upped now to 480 genes, as I mentioned. And the timelines, that's a 2-day, 3-day, and a 5-day timeline. So you can see the sort of trade-offs there. And that's, again, for 2 slides. Custom batches, so you can also order 1 to 50 or 50 to 100. It's a gentler cost point, and you can then scale. They've got a 4 reaction and a 16 reaction. You're going to pay more per reaction for a 4 reaction versus a 16. But again, their idea here was to lower the barrier of entry into the technology. Or for instance, if you find a prime data set that you're interested in, maybe you're fighting molecular crowding and things like that a bit, you could focus on a smaller set and extend your results from your initial findings. Proteins, RNA on the same slide. So they're targeting, again, this is first half of 2024. They're targeting 20 proteins at launch with the nucleic acids. It's oligo-conjugated antibodies. So this should expand very quickly once they get the concepts all worked out. Something that's coming, it's sort of end of this year, early next year, is improved multimodal approach to cell segmentation. So using nuclear membrane transcripts, cytoplasm to get a more concise, clearer picture. And the images there look really nice. So some advanced custom. So currently they have available isoform or translocation, so you can design custom probes. So here's an example in the top right corner, which again, I just need to keep doing this. You see the transcript at the top, and then you see the sort of little red padlock. So they use a padlock technology. The arms of the padlock are about, or I'm sorry, the binding positions are about 25 bases or so, 20 to 25. They're sort of ambiguous about that. But you can use those to discern differences. So you see the splice variant here between the yellow-green and the yellow sort of fuchsia color there. So you can distinguish those. Diverse species. So again, you can design your custom space. They ask for single cell data, as much transcriptome data as possible so that they can balance probe sets and things like that. But a key attribute that they asked us for was single cell data in particular to do that balancing. Xenografts, so you can mix your mouse and human together, for instance, again, in a custom flavor. You could use a fixed human panel with a mouse spike or vice versa, however you'd want to do that. Exogenous sequences, so viral, bacterial transcripts, reporter transcripts, things like that. And then in development, expressed SNVs, again, here's data demonstrating where they designed using a single nucleotide change at the ligation point. So one arm has the change and one arm does not. And you can distinguish very clearly the two transcripts here with the single nucleotide variant. TCR and BCR profiling, which I learned about yesterday, so I'm anxious to hear a little bit more about that. And then guide RNAs. So it's not on their roadmap, on their published roadmap, but they're getting pounded for this. There's lots of interest. And again, so one of the attributes of 10x that was appealing is the sort of landing spot of the padlocks. It's 20 to 25 bases. So you could imagine designing small guides to where you can hit the specific guide and identify it. Post-xenium processing, so H&E, IF, IHC, and then Visium. So these are all things that you can do after running the xenium. So the tissue is intact after running the platform. They already have software, and Rich will talk a bit more about this, doing the H&E afterwards. But they have software to overlay the H&E image, which you can see some of the, there we go. The overlay images on this. And then Visium HD. So this, again, is a significant appeal to us, where the Visium HD is similar to the Visium, but it will do near single cell. It's actually subcellular to some degree. They're looking at 2-micron grids that collapse into 8. So it does help them to segment when you get multiple cells sort of overlapping grids and things like that. But near single cell, and that's full transcriptome. So we envision sort of the utilization of both this and the Xenium and the Visium HD. And that's it. I'll hand off to Rich.
Okay, thanks, Bob. So yeah, Bob talked a good bit about what went into our decision process on what platforms to target. And I'm going to talk a little bit more now about, okay, we've made the decisions, we've started to implement these platforms, we're bringing them online or have brought them online already. in the MGI, and I'm going to talk a little bit more about the practicalities of what it takes to do these experiments, and some of the things that you need to know if you're planning on doing experiments with these platforms. So I'm going to go a little bit over the roadmap, which you've heard a little bit about already from Bob, and I'm just going to kind of reiterate a few things. Then I'm going to speak specifically to the Xenium, a little bit about Visium, and then I'm going to spend some time on the bioinformatics, because we've already been getting a lot of questions about the bioinformatics and getting help with processing data, et cetera, and some of the things that we're doing there to help give people a little bit of a leg up on getting going with the analysis of their data. Okay, so as Bob had mentioned, this is not the first time we've used a 10x genomics platform. We've been using 10x since the Chromium days for single cell, and 10x is a very good track record. They've delivered typically what they said they're going to deliver. Not always exactly at the time, but generally within a reasonable time frame. And most of what they've said that they would get to us eventually, they do. And so this was definitely a big factor for us moving forward with the Xenium platform. We brought Visium online last year. That was a relatively easy decision because it used most of the same equipment that we already have to run the protocol. Then Xenium was the bigger decision because these instruments are actually fairly expensive, and so we had to make a pretty good investment. That said, as we'll point out a little bit more as we go forward, there are ways in which these technologies integrate within the 10x family, and that was also an attracting factor. So as Bob mentioned, here's a little bit of the roadmap. We've already launched. We've launched all three of these now. Chromium's been around for close to 10 years, but we launched Visium, the low definition of Visium, last summer, and we just launched Xenium over the last few months. And as also was mentioned, there are additional panels being developed for Visium, additional predefined panels, and the proteins should be available roughly in the first quarter, around the time that we're expecting Visium HD. And I'm going to actually show we do have an image from Visium HD that I'm going to show to compare it with the current Visium, and it's pretty astounding, the difference. Okay. A couple things that I'm going to point out. A bit of this was mentioned already. This is a probe-based platform, high resolution subcellular. There's about 6 panels already designed, with 2 additional available soon, very soon. We've heard within the next week. One of the key things I want to point out here is we have 2 workflows that I'm going to go through in a little bit of detail with you, because there's some decision points you would have to make as a laboratory and how you want to do this. And it's a pretty important decision point, because as we'll get to at the end, this is not cheap technology. I'm going to just point that out right now and make sure you all understand that at the end. You don't want to have a lot of failures here, because this is not like having a failed run where you spent $100 on sequencing or something. This is a much more expensive platform. We have 2 FFPE workflows that are up and running, and this really just depends on whether or not you're going to make the slide or you're going to make the slide with us in the DDRCC. There is also a fresh frozen protocol in testing right now. Basically, when Jen's group tells me, yes, this looks good and it's ready to go, we'll bring that online as well. So FFPE you have today, fresh frozen, hopefully within the next week or 2, we'll be ready to pull the trigger on that. We'll have to see what the data look like. We have 2 Xenium instruments installed now, so we have substantial capacity. To the point that Bob made earlier, even with only a 2 to 3 day run time, as you can imagine, if you've got a lot of projects, it can take a while to get through. So we thought it was important probably to have 2 instruments in house, because if we had any sizable projects, that could really delay other folks that wanted to do smaller runs. So that's one of the reasons we decided to go ahead and get 2 instruments almost immediately. So when it comes to, and Bob mentioned the part about the pre-designed panels, I want to talk a little bit about designing your own custom panel, because there are some considerations here. So when we've talked to 10x about this, they've made it very clear, even when they do their design, they, on their publish, they say, yes, we've used information from Single Cell Atlas and researchers and experts and literature, et cetera, et cetera. But when you really get down underneath of it, they say they really like to find things that have worked well in the single cell or Visium space, because those have a really high probability of working. Those probes, or probes around those transcripts, have a higher probability of working in the Xenium space. And so it's good to find, if you're going to target certain genes, it's good to go to some of these single cell databases to see if probes like this or probes associated with those transcripts have been used successfully in the past, because that can actually be a factor in improving your success rate if you're going to do custom panel design. So that's really the reason I wanted to point this out. And yes, my numbers are so day before yesterday. Mine says 300 and now they're up to 480. So apologies for that. And you know, as you can see, this is a very rapidly changing space. The numbers of genes, the different things, the entities that you can measure, the molecular species that you can measure, et cetera, are changing very, very quickly and generally in your favor. Okay, so this is a visual, and I'm going to get more into the actual details in just a minute. This is just kind of a visual to give you a sense of what goes on in setting up your experiment and how you have to prepare your materials in order to be able to do this. So up above, basically you can see you have to, there's sort of a standard sectioning involved. And I'm going to talk about this number 4 in good detail next, because this is really, really critical. I cannot stress enough how critical it is that you place your sections properly on these slides, because if you do not, you could end up with a very expensive failure. Okay, it's really, really key that that part is done very well. And then the rest of this is really just kind of walking through the process of the Xenium, the deparaffинization, the cross-linking, et cetera. And then a little bit about the in situ process that goes on. Below here is sort of a breakout. You know, Bob mentioned 2 to 3 days for the runs. This is a little bit more detail. You can see that really your upfront is relatively small. It's about an hour, hour and a half. The run itself is on the order of a little over 24 hours based off of standard section 2 slides, clean up and unload. So the whole thing together, by the time you get done with the process, is on the order of about 2 days, depending on what you've done and how much of the space that you're using for sections. And with that space, it is certainly possible to put multiple sections. As you saw, this is 12 millimeters by 24 millimeters. You can put multiple sections in that space, but be very careful not to have those things overlapping or anything like that, because again, that can create problems. Okay, so this is a little bit more detail now in terms of what this looks like. So you are going to make a decision about the panel that you're going to run. You're going to use something off the shelf, you're going to do something custom, maybe a mixture of the two. You make that decision about what you're going to do. You get your, this is what we're going to do. We're going to run this panel, or we're going to do custom. There will be lead time if you do custom. So be aware, if you're going to do a custom, I think it's about 6 to 8 weeks. I think is what they're generally, they say a month now, but I would say anticipate at least 6 weeks for that panel. If they get it in a month, great. It's a pleasant surprise. But if you guesstimate 6 weeks, give or take a little bit, you should be in the ballpark. Now, if you're not comfortable doing the sectioning and the placement of the sections, this is one of the reasons that we have a collaboration. GTAC and at the MGI and the DDRCC are collaborating on this, because even we don't have that kind of expertise in our facility, but the DDRCC does. They do sectioning, placements of these sections. They are experts at this stuff. They have expert histology technicians that can do this stuff in their sleep. And so we have partnered with them on this. And so if this is not something you feel comfortable doing in your laboratory, again, given the expense of these experiments, we strongly recommend partnering with the DDRCC to actually basically get your sections and make your slides. Very, very much recommended. Now, there are some labs that have a lot of sophistication with that and are very comfortable doing it themselves, because they do a lot of this stuff. And we work a lot with Ben Humphrey's lab. Ben's lab is very, very good at doing this stuff. And in fact, we talk with them quite often because they're very sophisticated in this space. They've been doing single cell and spatial, and even in the bioinformatics field, we're kind of talking with them and comparing notes. So it can be done. But if you decide to go this route, then basically you would deliver your blocks to us or the DDRCC. If you deliver them to us, we'll take them to the DDRCC and basically go through the sectioning and placement on the slide. Then they come back to us. We do the deparaphrasation and the slide prep. We do the Xenium run. And at the end, I'll tell you a little bit about it. Some of the analysis that we're setting up is just standard analytical pipelines that will come out with the data by default. And then also there's an optional A&E, which you can, if you're not comfortable doing that yourself, you can also talk to the DDRCC about that as well. Now, if you do feel comfortable doing the sectioning and slide placement yourself, this is basically how you modify the protocol. Again, you still have to do all this stuff up front with the your panel review, decide what you're going to run, etc. You do the sectioning. Then basically the slides have to be baked for 3 hours and then desiccator overnight. And again, there's multiple notes here about things you should consider when doing this. And then at that point, you would deliver the slides directly to us. And in fact, if you want, you can even order the reagents directly, the slides, and get the slides directly delivered to you from from 10X if you prefer to go that route. If you're working on a large project or with collaborators, you can go that route where you can get the slides and the reagents sent to you and then bring us the slides for processing once they're ready. Or again, you can work with us for that part. And then at that point, it's relatively similar to the prior process. But again, really stressing, if you've got a lot of experience doing this, if you have people with good experience doing this, if you're comfortable doing this with us, that's great. If not, I really do recommend working with the DDRCC to get your sections and make the slides. Okay, so also, this is another one of the things that's been mentioned briefly, but this was another attractive factor to this. So 10X, in addition to having all of these platforms, the Site Assist instrument can actually help you transfer. If you would like to do a Xenium run, and then you say, wow, okay, this is great, but I need to know more. I really want to do a whole transcriptome run on this. The Site Assist can actually transfer the sections from the Xenium slide to a slide for Visium. And you can basically take those same sections and process them for whole transcriptome. So instead of having to go back and basically get new sections off of the block and put those on, you can actually process the same sections. So this was very intriguing to us, this ability to kind of go back and forth between the technology platforms. Now, at the moment, it's one direction. As far as I know, you can't go from Visium to Xenium. You can only go from Xenium to Visium. But even that, basically, we saw as a potential advantage depending on what investigators were trying to discover from these samples. And the Site Assist can also be used just for the transfer process itself. If you basically want to put your sections on a plain glass slide, we can use the Site Assist to actually transfer those to the Visium slides for processing after that. Okay, so I'm not going to go through this in a lot of detail. This is the Visium workflow, or the basics of the Visium workflow. What you'll actually see is they're not all that dissimilar from the Xenium workflow, at least at a conceptual level. You go through a lot of the same processes and steps, or similar steps, in order to get to the part where you're doing the run. The other big difference here, though, is Visium is a sequencing readout. So with Xenium, you get your readouts directly off of the instrument. With Visium, after you've done this run, you're actually going to take those slides, you're going to make libraries from it, and you're going to put those on a sequencer, on an Illumina sequencer. And as probably most of you know, we have the new NovaSeq X sequencers. So this can be an expensive component to Visium if you want to sequence deeply. We haven't really done enough testing to tell you, is this more like FACSeq or is this more like a 10X Chromium type run in terms of the limitations of the numbers of transcripts you can detect per cell? So we haven't done like a deep sequencing run yet on Visium. We were kind of waiting for Visium HD for that. But depending on how deeply you sequence, that can be a little bit more expensive component of this. With the new with the new Illumina NovaSeq X Plus sequencers, and we're expecting the 25B kits, which are really bringing the price way down, probably within the next month or two. We think Illumina still hasn't given us a formal date yet, but we're hearing rumors that it could be within the next month or two that we'll see those 25B kits, which will really help with the sequencing cost as well. Now, You say, what's the difference between the current Visium and the Visium HD? Well, up here, you're going to see a really, really big difference. So over on the left, this is I say about 50 microns, it's 55 microns specifically are the spots. And I've really blown this up, but you can see over here that this is mouse ileum. And where you see these orange and green dots, you'll see maybe about 2 to 3 dots total. Those are the individual villi, or the villus structures in the ileum. And the orange that you see there represents the, sorry, this one's kind of hard to do. Okay, I'll just point to it this way. The orange dots represent the enterocytes. The green dots on that slide represent the basically like the progenitor cells. And then underneath of that you probably can't see too well are the red dots are where your crypts are at and where your Panna cells are at. And then below that, you start getting into fibroblasts and plasma cells. Over here, you can see how much better the resolution is on the HD. So what you see over here is the villus structure. You can actually even see, I don't know if the battery's not working. You can actually see where you can see the immature epithelials moving up out of the crypt until you get to the mature epithelials at the top of the villi. And you literally just don't pick that up on the current Visium platform. So, and you can't really probably see it back there, but those tiny blocks are the pixels that you're picking up. And I suspect those are probably the 2 micron sections that they're referring to. So while it comes by default, they've told us at 8 microns, you can, if you want, if you feel comfortable doing it yourself, partition it down to 2 micron resolution. So quite a bit of an improvement in terms of the resolution and what you can get out of the Visium HD platform. So a lot of people are going to ask the question, well, okay, which way should I go then, right? I mean, if I can get that resolution with whole transcriptome, well, that's not subcellular. So let's be clear about that. But that's something you're going to have to weigh as well. What will make more sense? And that's honestly where I think, if you're thinking through the experiment, contacting us, we can give you a lot more information that may help you make a decision about what would be a better route to go. Going with a custom panel or a pre-designed panel for Xenium, or going with something like a Visium HD. Now, keep in mind, Visium HD won't be out for about another roughly 6 months, we're hearing it, sometime in the first half of 2024. So, targeted versus whole transcriptome, higher resolution, lower resolution. Here you do have to go back and sequence. So there are a number of factors to be considered. Okay, now what about all the analysis stuff, right? So we probably get this question as much or more than anything right now. Interestingly, even externally. So some of the pharma companies that we work with are very much generate our FASTQ files and send us the data because we have to process it here. With this platform, we've been getting asked by the pharma companies, is there any analyses you can provide for us? Because they literally don't know what to do with the data. So we're in the process of setting up some pipelines, some fairly robust pipelines to get people going. There are software that come with the platforms. And some of the things that they do, some of the images that you can do, the cell segmentation is getting much better with some of the 10x software, but it is pretty limited. There's just not a lot of stuff that you can do with it yet. Now that may change over time, but we're not going to wait. We're going to go ahead and get some of these workflows up and running in order to be able to deliver a much more usable product to you for your research. So right now with the Xenium run, there is analysis software that comes on board that can generate a certain amount of information for you. But we're also now incorporating 2 different workflows that are available in the public domain, Seurat 5.0 and SquidPy, which are both set up to be able to take the output from the instrument in as input. And they give you, there's some overlap, but there are some different flavors in terms of what you get out of the 2, and I'll show you that in a minute. And then basically we'll just deliver those data to you with the underlying data files and data that come off of the instrument. So basically you would get a data package that comes back from us that would include all of those components. Visium, that will be next on our roadmap, and we'll probably release workflows we'll see. We'll see if we're able to get to that because there's a lot more we're going to do. As I'll show you, there's more we're going to do with the Xenium platform in terms of the software. This is just to get things started. And we have quite a bit more that we're going to be adding into the system. But we will probably try to align a release for Visium analysis also around the time of the HD, which, as we mentioned, may only be about 6 months out. Okay, so Seurat 5.0, as I mentioned, it can read and filter the cell and gene data from the onboard software. So that's nice. It actually integrates fairly well. It will do your standard data transformations and normalization, clustering, dimensionality reduction, and so forth. You can get UMAP plot creation out of it. And basically, it integrates very nicely with Xenium Explorer. So the nice thing about Seurat 5 is it's already designed to plug in pretty well with the Xenium platform and basically what comes off of the platform directly. So this is nice, and it can actually help get you a lot of information associated not only with the clustering of your cells and the information, but some of the visual data as well. The SquidPy pipeline is nice in that while there's a little bit of overlap, there's actually a lot of QC information that you can get out of SquidPy, and this is one of the reasons we're adding that into the workflow. You can see this up above. I'm not sure how readable that is in the back of the room, but transcripts per cell, unique transcripts, area of segmentation, these distributions you can get relatively easily, clustering coefficients, centrality. These are all sort of what I refer to as more of the QC measures that associate with your data. And SquidPy is actually pretty good at generating this. And again, with work from Gusu and Marie from our analysis team and really the whole team, which I should have mentioned a little bit more. We have 8 bioinformaticians working on this right now to get these workflows up and running so that people will have something ready as soon as they do their experiments, as well as spatial graphs. So this is not the end-all be-all by any stretch of the imagination, but this can generate a lot of information for you to really get rolling with your data, especially in these early days where there's not a lot out there. Now, beyond this, we have multiple things planned. So this is what I just showed you is just phase 1 of our analysis plan for these platforms. And that's pretty much about ready to go. We estimate that we'll be releasing this by October 1st, which is about a week and a half away. The Seurat 5 is already ready to go. The SquidPy is getting very close. We hope that'll be ready to go within the next few days to a week, and we'll be releasing all of that along with the full data package that would come out with any of the data that we generate off of the system around the first week of October. Phase 2, and this is where we're working a lot with Ben, so basically speaking with the folks from Ben Humphrey's lab as well as Jeff Milbrandt's lab. We're doing some more sophisticated work and pipeline development around this, and actually doing some methods development around this. And so we're in communication with them. And I must say, Ben's been fantastic. For those who don't know, Ben Humphreys is very, very sophisticated with single cell technologies as well as spatial. His lab has done some great work, and I think there's a pipeline that they're publishing right now, or at least the paper's in review, I think. And so we're working closely with them, and he's been very, very gracious in sharing what they've learned. And so I just want to kind of put that out there as well as Jeff's lab. I should mention Jeff's lab, obviously, because they're doing similar things in the neuronal space. So we're trying to work as a community on this, and with the labs that are fairly advanced with it, as well as the fairly substantial resources we're throwing at it to try to deliver workflows that can get you a lot of what you need. You may still want to do a lot of custom analysis at the end of the day, but we're going to try to make sure that you've at least got all the basics in hand out of the gate, and you're going to have what you need in order to get going. And then finally, the phase 3 development, and this is the sort of thing where some of this we might be able to incorporate from the outside world, and some of it we may be developing ourselves. And this is where we're going to be working to cross over these different platforms and integrate across them. So integrating single cell data with spatial data, and we even have a plan to try to integrate and project and infer with bulk data. Because as you probably know, all of the single cell and spatial work, you generally don't see all the transcripts present. There are limitations, especially with like droplet technologies. If you run single cell analysis on something, you might get 5,000 or 6,000 transcripts. We know that if we do things like FACT-seq or SMART-seq, you can see 12,000 transcripts in those same cells if you sequence deep enough. And so we want to try to basically make it so that you can, I saw somebody had a picture the other day of like a milkshake with bulk versus like these individual components for these single cell and spatial, which is true, but with the bulk, you can actually get more about what ingredients are there. So we're going to be doing some work to try to integrate those two things more directly so that we can actually make projections about what's going on in the bulk data. So as you can see, we're putting a lot of effort into the bioinformatics analysis. So summary of all of this. As you've seen, we've made a pretty substantial investment in the 10X platforms. That's not to say we're not going to keep our eyes on other stuff. We're absolutely going to keep our eyes on other platforms. We're not married to 10X by any stretch of the imagination. So if there are things that come out, we will look at those. Xenium is online for FFPE. That's in production. Fresh frozen's not far behind. Proteins are on the near horizon. Visium FFPE is online. Currently the low resolution, but high resolution is on the not too far out horizon. And for the bioinformatics, we have some robust pipeline development that's almost ready to go. And then more advanced analytics and integrative analytics right behind that. So we've got about 10 minutes left. So I'm going to stop here because I'm sure there's going to be a reasonable number of questions. And I'm sure the first question is going to be about what's the cost. So, okay, so I'll stop there. And Bob, if you want to come up so we can answer questions, we'll open it up. Thank you. Never would have thought somebody would ask that question. So to put it, yeah, so obviously the two platforms are a little bit different, but the reagents for them kind of run roughly in a little bit over the $6,000 ballpark for 2 slides worth of run. So I think our full in cost is about $7,500 total. And that includes, you know, all the prep work, and I think that even includes the DDRCC component. So it's about, and now that's basically 2 slides worth, and then however many sections you can get into those 2 slide areas. That's internal. I want to make sure if there's anybody from outside, depending whether you're profit, not-for-profit, they can go up another 20-25%. But internally, on the order of about $7,500 for 2 slide run.
And you guys sell some of the standard panels they have, like
well, you can, yeah, I mean, standard panels, we just order when you, and that's actually part of that cost.
Yeah.
Now, if you're going to do a custom, that's a little bit different, and we can get into that if you want to do a custom panel, because it's too much put up here. There's different sizes, there's different reaction numbers, and all kinds of stuff like that.
And it's evolving quickly. So 10X has, like I say, yesterday, these small custom panels options and things like that to lower that barrier to some degree. So it's evolving.
That one's better handled as a conversation.
Yeah. So,
helps. So Visium is untargeted. Visium is whole transcriptome. Xenium is targeted. Xenium is all targeted. Yes. Visium is whole transcriptome. It's untargeted. Just work away across the room here.
in the middle. Right.
I'm sorry, I didn't quite catch the. Oh, you want to?
Again, that would be it'd be a custom add-on, custom probe set, much like you would target a human transcript, for instance. You could target the same, your reporter gene transcript.
The processing is the same.
Yep. Yeah, it'll be, those probes will be integrated in amongst the or potentially you wouldn't have to, but you could have a pre-designed panel. You could integrate 50 customs for reporter genes or whatever it might be and target in parallel.
The Xenium is subcellular, whereas even the Visium HD is about 2 microns. So it's getting you down on cell level. I mean, whether that's subcellular, I mean, it's still, I would say, roughly an order of magnitude less resolution.
Against the wall in the back.
They have mentioned slide tissue types that can be difficult to work with. And I don't know if they have like a published list or not, but they have mentioned to us in some of our discussions with them that there are some tissues that are a little bit harder to work with. I would say if you've got a question, we would directly send that a question to 10X to say, here's the tissues we've got investigators thinking about. Are there any in here that you guys know that are particularly difficult to work with?
And 10X's website, again, it's second to none, in my opinion, in terms of help for that sort of thing. The technical support is great. Yeah.
I have two questions. For the first time, how
Do they validate the probes? No, I don't think they validate them. And that's why, if you remember, I was stressing, basically see if you can identify probes that have worked in the single cell space or other spatial work, they actually recommend using those because those tend to work at a fairly high efficiency rate. If you get outside of that space, you run the risk that the probes may or may not. I mean, their design is good, but it's always good if you've got data somewhere that shows that those particular probes have worked in the past.
One thing I think that worked in that thought process, one thing that didn't occur to me to begin with was if you design a panel, for instance, you want to think about that through all your perturbations you plan to do as well. And 10X walked us through a lot of that process where, because it changes the gene expression significantly. So that's something that you can work with them to keep in mind.
We've only got a limited number, so I thought I had the numbers in here. We've run 7 projects so far on Xenium. We've run 20-something projects on Visium, but it's not been a huge swath of tissue types. So obviously we have tested in the space that we've worked, and so far what we've seen, especially the new workflows that we've been looking at, and with the latest software from Xenium, the cell segmentation is quite a bit better than it was before.
Yeah, and it's coming, the new advances in the segmentation are coming probably beginning of next year. Not at the moment. That's come up also. So we had that, I had the discussion with them yesterday. So not at the moment, but in the thought process, it's challenging. Anybody else?
Taking into the Xenium process, you diminish the signal a lot. In other words, you're putting this one code onto the RNA. I guess the code is turn it off, and then it would go into the Visium, and the RNA is unaffected by this whole journey.
We haven't tested it ourselves. That's what we've been told by the manufacturer, is that you can go one from one to the other and there's no degradation of sigma, or at least nothing significant.
The idea would be to take one food, don't take it to make sure.
You could do an experiment like that for sure, but yeah. Sorry, I couldn't quite hear what was saying here. Well, so a couple things. Basically, the alignment is sort of key, and especially making sure that it aligns well with basically how you've done, like if you're going to do an H&E or something like that for overlay. And then basically there are these there are points, basically alignment points on the corners, and you got to make sure you keep things within that. If anything goes out, it's a problem. If you put multiple sections on there and there's any crossover, it gets a lift, and that creates all kinds of problems. So there's really a number of different factors depending on what you're trying to do with the slide. And so it's just like I said, if it's something you're comfortable doing, and you're pretty comfortable, you've got some relatively small section, it's going to fit in there, no problem. You're comfortable, making the sections and putting them on slides for microscope work or whatever, might be just fine. But if you don't have that ability within the lab, or, you know, especially working here, like I said, making sure you're sort of in the neutral zone of the slide, because if you get outside of that, it creates all kinds of problems. Site Assist that's not software, that's an instrument. It would actually take the section from a slide from Xenium and it'll transfer it to a slide for Visium.
It actually just transfers the nucleic acid.
Oh, right, yeah, sorry.
It lines it up and
it transfers the nucleic acids with the aligned. But yeah, I mean, you basically get it aligned as if you had the same section of tissue on both slides. But yeah, it's not software. That's a physical instrument. You basically put the slides in and it does the transfer. Which platform to choose for the best spatial information? It depends on the problem that you're trying to solve. So the two platforms have different strengths. The Xenium is higher resolution. You can get down to subcellular level where, Bob showed you can really see almost down to the transcript level. But it's with a fixed transcript set. So you're always working in a fixed space. So you kind of need to know what you're looking for or within a certain region. Whereas Visium is different. Visium says, okay, basically we're gonna tag all the RNA that we find in there, and then we're gonna make a library out of it, and we're gonna go sequence it. And then we'll tell you where that came from on the slide. And so it's basically doing a whole transcriptome level analysis, as deep as you can go with these types of technologies, and telling you everything that it's finding in those spaces, but it's lower resolution. In fact, right now it's very low resolution. You get these 50 micron, 55 micron spots. The HD will be much better. It will get you about to cell level resolution. So I'd say if you're debating, if you're thinking about doing something, you're debating, it'd probably be good to just sit down and have a conversation with us, and we can walk you through. If you kind of tell us what you're trying to identify, we can probably walk you through what would be the best technology to use to do that. Yeah, the costs are, by the time you add on the sequencing and stuff, yeah, the costs aren't they're not too different. Unless you're going to sequence really deep, then actually Visium can get more expensive, but only if you're going to sequence really, really deep. Because that's kind of a variable component to the cost for Visium. That's a very good question, and I'm not sure. I mean, it tends to be the better expressed transcripts, but it's not always the case, right? I mean, we see this with single cell too, right? I mean, you go in with single cell, and I don't care how deep you sequence with Chromium, you're going to pick up 5,000-6,000 transcripts depending on the cell type. And most of the time, those tend to be higher expressed transcripts, but not always. Some that, for whatever reason, there are just certain aspects to this. It's kind of a little bit of almost like a quantum black box, if you will, as to why, and I'm just using it as an analogy, as to why some transcripts picked up where others don't, but they tend to be the higher expressed transcripts. And it'll be the same thing with the spatial. It will tend to be the higher expressed transcripts. So if you're looking for a lot of like lowly expressed transcription factors, you might be better off looking for the things those transcription factors activate and drive, because those signatures will probably be easier in a lot of cases to pick up than the low-level transcription factor. Not always. Sometimes you do get transcription factors, but you're not going to necessarily count on that. If you know what you're looking for, yes, because there your chances of picking it up are much better.
And 10x tailors the probe sets based on the gene expression also, so as to not swamp out some of the lowly expressed transcripts.
Yes, actually, we'd recommend it. You know, give us a call. We'll be happy to sit down and have a conversation. Sorry. On problems on, I'm sorry, tissue microarray. I'm not sure I followed the. I'm not sure entirely if I followed the question. So you have a. Right. Yeah, I mean, I mean, if you're taking sections from individual patients and you're processing them, and then you're basically doing a data analysis. So are you asking, can you connect these data back to what you get from tissue microarrays? Or I'm sorry, I'm just not quite following. Oh, yeah. I mean, yeah, if you're getting this, yeah, the sections, the same types of sections, you wouldn't be able to obviously use the same section, but the same types of sections cut from the same block or whatever, that should be fine. Yeah. Sorry, I apologize. I didn't quite follow the. Those were both Visium, but one was Visium, one was Visium HD. Sorry, it's kind of hard to hear from the back of the room. Would it be the same?
The thickness.
Thickness? Oh, same thickness?
I think.
Yeah, I think they're the same thickness.
So, yeah.
And if I recall, they were, don't quote me on this, but I think they were even saying that Visium HD will be roughly the same price as Visium, won't it? Or
Yes.
ballpark, or pretty close, I thought.
Sequencing will be more.
Yeah, sequencing, but that's the variable part. I
That's
mean,
the
the 10x component, I think, would be
component
roughly
wise.
the same. And then the sequencing, again, depending on how deep or shallow you might want to sequence, that'll be. But yeah, I think the thickness is the same.
Yeah, I think 5 to 10 microns.
5 to 10
correctly.
microns is the, yeah.
Yep.
Yeah, I don't know why it wouldn't.
Yeah, I've seen
Yes.
they're, and I need to follow up with 10X. They actually sent me a slide where they've actually got a grid of different cores that they gridded out, recut, put down. Well, it looks great.
And that actually almost seems like a better way to do it, than trying to take a section from each one and lay them down nicely and not get anything overlapping.
Yeah.
We good on questions? Good
Oh.
thing we left about a lot of time for questions.
Exactly.
All right, thank you, everyone.